All right, class. So, talking about different phases of gastric secretion. So, when we say gastric secretion, we are actually talking about stomach secretion. There are three main phases that we will talk about, and I have written down all three phases here. The first one is cephalic phase, which, as the name suggests, is controlled by the brain. The second one is gastric phase, which is controlled by the stomach itself. And the last one is intestinal phase, which is mainly controlled by part of the small intestine. I want you to know that the cephalic phase and the gastric phase, they increase gastric secretion. And the intestinal phase decrease gastric secretion. And when we say these two phases increase gastric secretion and the intestinal phase decrease gastric secretion, we are looking at a normal, healthy individual. Okay, so let's start with the cephalic phase first. Sorry. Okay, so when we go to, let's say, OC Fair, the smell of food, will that ever make you hungry? Do you ever feel hungry by the smell of food or the sight of food, or even like thinking of food or talking about food? I do get hungry right after a meal. If somebody is talking about some delicious food, I feel hungry again. And this is the function of the cephalic phase. Now the idea is these sensory stimuli activates the vagus nuclei in the central nervous system. And the vagus nerve in turn controls the cephalic phase of gastric secretion. The vagus nerve innervates the stomach, and the vagus nerve controls it in two ways. It can have a direct effect or an indirect effect. Directly, the vagus nerve can stimulate the chief cells to produce pepsinogen. The vagus nerve can also innervate the parietal cells to release hydrochloric acid. And indirectly, the vagus nerve can activate can stimulate the G cells to produce gastrin, which in turn stimulates the ECL cells or the enterochromaffin-like cells to release histamine. The histamine works as a paracrine regulator and causes the parietal cells to release hydrochloric acid. So as we see, the vagus nerve that innervates the stomach controls gastric secretion both directly and indirectly. I want you to know that the indirect effect is more dominant as compared to the direct effect here. Next, we have the gastric phase, and the gastric phase is stimulated by the physical contact of food with the stomach wall. There are two major stimuli for the gastric phase. First, distension of the stomach, meaning as we have more food in the stomach, more distension of the stomach, more chyme. All right. So yes, more chyme, more distension of the stomach, and that causes more secretion, gastric secretion. The second stimuli is actually the chemical nature of the chyme. So what is there in the chyme? If there are more partially digested proteins, that can activate G cells of the stomach to release more gastrin, which then triggers the ECL cells to release more histamine, which then works on the parietal cells to produce hydrochloric acid. The partially digested proteins can also work on the chief cells of the stomach to produce pepsinogen. So again, for the gastric phase, there are two stimuli: the distension of the stomach can cause more gastric secretion, so physical contact of food with the stomach wall here. And then also depends on what is the chemical nature of chyme. More partially digested protein, more gastric secretion. Okay, so as more hydrochloric acid is produced, obviously that results in a drop of pH, stomach pH, and when that pH drops below 2, the D cells are activated. And what are the D cells producing? They release somatostatin, remember? Somatostatin, it inhibits gastric secretion and function, so we see a negative feedback loop here. Moving on to the third phase, which is the intestinal phase. Intestinal phase is nothing but inhibition of gastric activity, and this takes place when the chyme moves from the stomach into the first part of the small intestine called the duodenum. Now for intestinal phase as well, there are two stimuli. First, duodenal stretch, meaning as more chyme moves into the duodenum, that activates the vagus nerve. The vagus nerve in turn produces a neural reflex that inhibits gastric motility. And also, if the chyme is rich in fat or fatty food, that stimulates release of few hormones. And the first hormone is somatostatin, and that is released from the stomach and the intestine. Then cholecystokinin, which we talked about before, and GLP-1. These are the three hormones that are released in response to presence of a lot of fat in the chyme, and these hormones definitely works to inhibit gastric secretion. Next, this is the abbreviated version or the chart showing the three phases. It pretty much summarizes what we talked about in the last 3 or 4 slides. Okay, so let's talk about absorption now. I want you to remember that absorption of most of the nutrients take place in the small intestine. Now, when we talk about carbohydrates, we take in carbohydrates in the form of potatoes, pasta, rice, which are starch. And also we take in other types of sugars like sucrose and lactose. As we saw in lab, starch digestion starts in the mouth or oral cavity with the help of salivary amylase. But remember, salivary amylase helps in partial digestion of starch. So then the partially digested carbohydrate molecules move to the stomach. And I want you to know that when the food goes to the stomach, there is absolutely no carbohydrate digestion in the stomach. So we have carbohydrate digestion in the mouth by the action of salivary amylase. We have no carbohydrate digestion in the stomach because it's too acidic. However, as the chyme passes to the duodenum, there are brush border enzymes that help breaking down sugars into smaller and smaller units. Also, we have an enzyme called pancreatic amylase that helps to break down the carbohydrate chains into smaller sugar units. Therefore, to review, carbohydrate digestion starts in the mouth by the action of salivary amylase. No carbohydrate digestion in the stomach. When the chyme moves into the duodenum, carbohydrate digestion continues by the effect of two enzymes. First, pancreatic amylase, and secondly, there are brush border enzymes like maltase, sucrase, lactase that break down sugars into their simplest forms. Moving on to protein digestion and absorption. Protein digestion obviously starts in the stomach by the action of pepsin and hydrochloric acid, and this leads to production of shorter polypeptide chains. However, protein digestion does continue in the duodenum, and jejunum by the action of several other protein digesting enzymes like trypsin, chymotrypsin, elastase, carboxypeptidase, and brush border enzymes. However, for testing purpose, I do not expect you to remember all these names. Just know that trypsin and chymotrypsin, these are the two main enzymes that facilitate protein digestion in the small intestine. And all these enzymes together help to break down the polypeptides or the shorter peptide chains into amino acids, which are the ultimate monomers that will be finally absorbed by the bloodstream to be transported to different cells of the body. Okay, fat. Fat digestion is a little bit tricky because fat needs to be emulsified first. We did talk about bile before. So bile does not help, bile does not work on fat, or bile does not break down fat. Let's put it this way. Bile emulsifies fat, meaning it coats the fat molecules or the triglyceride molecules. And when the triglyceride molecules are coated with bile, then the enzyme lipase acts on the triglycerides, breaking it down into fatty acids and glycerol. So again, fats or triglycerides need to be emulsified first by the bile. So bile emulsifies triglycerides. After that, the enzyme lipase acts on the triglycerides, breaking it down into fatty acids and glycerol. Now here, this picture explains or talks about the absorption of fat in the small intestine. So to start with, we see the lumen side of small intestine in yellow at the bottom. We see a lacteal, and we know that lacteals transport dietary lipids to circulation, to the venous supply. So let's look into the lumen side of the small intestine. We do see triglycerides there. And we also see that the bile salts emulsifies these triglycerides. And after that, the lipase works on the triglycerides and break it down to fatty acids and monoglycerides. Now, the fatty acids and monoglycerides also formed mixed micelles with bile and other bile salts. And ultimately, fatty acids and monoglycerides are taken up by the intestinal cells, as you can see. And when these molecules of fatty acids and monoglycerides get into the intestinal cells, they combine together to form triglycerides again. These triglycerides, along with other proteins, form structures called chylomicrons, which are then transported to the which are then picked up by the lacteal, and the lacteal then transports these molecules to the thoracic duct, which then ultimately dumps all these molecules into the venous circulation. So that concludes my lecture on digestive system. This was a super short video that I made. If you have any question on any of these, feel free to email me or talk to me during my Converse Zoom session next week. Again, this material will be covered, or you will be tested on this material in lecture exam 2, the end.